be familiar with the terms at the top of the outline. Most of them are commonsensical and you will already be familiar with them. This graphic is showing normal flora and bacterial counts at certain body parts. If you take a look at the mouth as well as the GI tract, you'll notice the numbers are pretty darn high. So here is that chart again that you have on your outline. This was listed as number 1 for portal of entry, and then with adherence and ID 50, they were both listed as number 2, and I'm bringing up the numbers because I added them throughout the PowerPoint. Then for damage to the host cell, that is your number 3, and then the last one, number 4, is actually portals of exit. Remember that these are very similar to portals of entry. So we'll start with portals of entry / exit. If you take a look at this picture, there are many different ways that pathogens can be communicable and infectious and passed on from one person to another, or just picked up by the individual that becomes ill. We'll look at the picture on the right. Sneezing as well as coughing, when it is a respiratory infection, is definitely a portal of entry as well as exit. Pathogens can be picked up in the urine as well as in the blood. Lesions are always an issue post-operatively. That is the biggest fear for potential of infection. Pathogens can be found in fecal samples as well as passed on through insect bites, oftentimes the vector. For example, think of malaria and some other illnesses, or West Nile could be something as simple as mosquito. The skin, which is not only the largest organ in the body, but it's our biggest protectant. And again, post-operatively, cutting into the skin to allow an opening to the internal portions of the body is incredibly dangerous when you think of infection. So within the skin, there are many aspects that make it a protective mechanism for us. First and foremost, we have hair on our skin, which just like in your nose or in your ear tends to catch different particles. There are sebaceous glands in the skin, which give it a basically oily-like substance on the top. There's keratin that is also a protectant. The skin has many layers, so there's the epidermis, the dermis, and the subcutaneous layer. So this is a very large protectant mechanism. This has Staphylococci written up here because Staph aureus is a bacteria that is a normal flora grown on the skin, or just growing on the skin. The skin has an optimal pH, which keeps the normal flora in balance and in check so that other types of organisms don't grow. When you have a skin or epidermal fungal infection, traditionally in that area, the pH of the skin is actually a bit off. So then when we look at number 2 on the list that's on your outline in the beginning of this PowerPoint, after portal of entry, the next thing to consider is ID. ID is short for infectious. Infective is another term if you're reading, but infectious dose. There's also an LD, which stands for lethal dose, but we don't spend too much time talking about that. What I need you to understand about infectious dose is a textbook definition. The number 50 actually refers to when you take a dose of some type of infectious organism injected into experimental subjects, and when 50% of that experimental subjects becomes infected, that is the ID 50. So you can read through letters A through E talking about how you have portal of entry, what happens with the host defense barriers, and then how you have portal of exit. But again, for infectious dose, I need for you to know that the 50 for ID 50 means that this is the amount of the infectious organism that is given to, subjected to, or ingested, breathed in, whatever that portal of entry is, by the experimental subjects or the patient, so that 50% of those subjects actually become ill. If you take a look at the bottom here, I just want you to please focus on the one in the middle. So you have young males that volunteer to determine the ID 50 of Neisseria gonorrhea. Gonorrhea is an STI, or sexually transmitted infection, and it causes urethritis. Itis is an inflammation of, so this is an inflammation of the urethra. If you take a look at the middle example right here, 10 subjects were given 1,000 bacterial cells, and 5 of them developed this inflammation of the urethra. This is a prime example of ID 50 because there were 10 subjects, and the 50 means that half of them became infected. So in this case, 5 of them developed the illness. Take a look at this chart. There is a unique relationship between ID 50 and virulence. Virulence, again refers to the severity of the disease. So something like lactobacilli, which you can find in basic yogurts like Dan Active, is considered less virulent, whereas Yersinia pestis and the Telornisis, these two are considered more virulent. So the relationship between ID 50 and virulence is the lower the ID 50, the higher the virulence. These two are what's known as inversely proportional. When ID 50 goes down, virulence goes up. A simple way to understand that is with lactobacilli, that would be a whole lot that would need to be ingested or go through a portal of entry for a person to become infected, as opposed to, say, Bordetella pertussis, which causes whooping cough, or Yersinia pestis, which is the plague. You need very little bit or a tiny small amount, and you already have a high level of increased virulence. So please remember that ID 50 and virulence are inversely proportional. So now we've moved on to number 3, which talks about several different things. How to penetrate and evade the host defenses. So what can these organisms do to invade the host? Because we do have an immune system, if you're thinking about humans, and we don't become ill every time we come in contact with some type of microbe. Another aspect to number 3 is how the host cells are actually damaged. And then this is all pulled together, if you look at the chart on your outline, by colonization. So one aspect of that is blocking phagocytosis. So phagocytosis is the ability to remove pathogens and debris through an ingestion. So you have a phagocyte, which we will talk about much more in the second half of this unit, part of your immune system. When a bacteria enters the body, I'm going to focus on bacteria, it can actually ingest it and then essentially chew it up and spit it out. This is a protective mechanism. Phagocytosis, the way I explain it, if you can remember the video game Pac-Man, it goes around the body. We have them all over our body, circulating throughout our blood, chomps up the bad guy, and spits it out. An example that I want to mention that has to do with evading the host defenses. Many of you have heard that if you have a UTI or urinary tract infection, that you should drink cranberry juice. But seeing how that actually works, most people can't really explain why. They just say it's good to drink cranberry juice and it flushes out the system. So let's take a look at why. Bacteria often have pili, and what pili does, remember when we talked about conjugation, they're sticky and they stick together. That increases the chance of a biofilm or protection in numbers for the bacteria. It makes them a little bit more virulent. So in this case, in the picture on the left, you're looking at a lumen in the urethra, and through urination, the bacteria lacks pili, and it just goes right through the urethra and urinated out. Now in the lumen of the urethra, when you have bacteria that have pili, notice they are starting to stick together. That can cause some type of blockage. So cranberries have the ability to block adhesins. Remember, adhesins are the sticky factors at the ends of pili that connect those two bacteria. So if cranberry juice has the ability to block adhesins, it will then prevent them from bundling together and bunching together and blocking off that urethral opening. This then brings us to the next portion of number 3, where you'll see that there's colonization written on the bottom and kind of connecting them together. So some pathogens invade cells and some pathogens invade tissues, but it is important to remember that bacteria have a tendency to mostly invade tissues. When you hear colonization, think of colonizing or congregating and grouping together. The picture on the top as well as the picture on the bottom are exactly the same. They're just taken from different places, so I'm going to focus on the one on the bottom. What you have is epithelial or skin cells. And notice here, it says basement membrane. If you think back to A&P, a basement membrane is important because this is closest to the bloodstream. And you should acknowledge that when a pathogen, any bacterial or any type of pathogen touches the bloodstream, that can take an infection that might start localized in one area and turn it into systemic. So the goal of the host cell's defenses is to make sure that that doesn't happen. So when a pathogen attempts to invade epithelial cells, it can release an enzyme called hyaluronidase. And notice what it does is it's breaking down, so there it's the pathogen is producing this and releasing it, is breaking down the host defenses and those tight gaps between the epithelial cells, allowing the pathogen to get deeper and deeper until it touches the basement membrane. Again, why is this a problem? When you get into the basement membrane, then is the time that an infection can become systemic. An example of that is Pseudomonas aeruginosa in lung tissue. We've talked about biofilms before, so I'm just going to bring that up again. Pseudomonas overgrowth, which is a normal flora and lives within our nostrils, our lungs, and many places in the body. When it overgrows in the lungs, that is one of the issues when someone has cystic fibrosis. So again, building of biofilms, we've talked about this in the past in terms of UTIs and when medical devices such as catheters are involved. Pseudomonas aeruginosa in the lungs is another example, and this can often lead to more chronic infections by breaking down the host defenses. So another example of tissue invasion is right here on the bottom, related to invasins. What I'd like you to look at is here you have a bacteria, and notice it becomes cradled within the cell of the host and then engulfed. It's almost another type of phagocytosis going on. There can be invasion of cells, although I did mention before that that's not really normal, and traditionally bacteria tend to invade tissues as a whole.